types of vaccines. In this session, we would consider macromolecules, purified macromolecules as vaccines. So these kind of vaccines were developed with the purpose that they would be safer vaccines. So let us look at the learning outcomes of this session. The antigenic determinants on the pathogens are immunogenic, and these are mostly macromolecules such as polysaccharides, glycoproteins, proteins, etc. So it is always these surface molecules that act as what we call as pathogen associated molecular patterns, and these are recognized by our immune cells, and therefore the immune cells can elicit an immune response. So with using the whole organisms, there is a risk of getting infected. However, using these antigenic determinants cuts the risk of infection. When you use just the surface molecules, you would not have the whole organism, and therefore it would cut the risk of infection, and that is an advantage. But the immunogenicity still remains. A mixture of macromolecules from different organisms can be used in a single vaccine to develop immunity for more than one disease. The most common macromolecules used in vaccines are exotoxins, capsular polysaccharides, recombinant microbial agents, etc. We will first consider the bacterial polysaccharide capsules. So say for example in Klebsiella pneumoniae, on the surface is a capsule which has what is called as the capsule polysaccharide. So you can see over here, these are the CPS, which is capsular polysaccharides, and these are basically molecules that can be antigenic, and they are recognized by the immune cells as antigenic. So what would definitely happen is that these capsular polysaccharides can be bound to by the complement molecules, say for example C3b and antibodies, and because of which you have an immune complex being formed, and these immune complex can be eliminated by activating the complement mediated lysis, forming what is called as the membrane attack complexes on the Klebsiella, and the Klebsiella will therefore get lysed and destroyed. Or what can happen is that when you have the C3b or the antibodies bound to the microbe, then these are going to act as opsonins. So they are going to recruit more of phagocytic cells, and which would engulf it, and of course through phagocytosis destroy the microbe. So therefore the understanding is very clear. If one uses just the capsular polysaccharide, you will again have the antibody CPS complex being formed, or you will have the complement capsular polysaccharide immune complex being formed, and that itself can elicit an immune response. So instead of using the whole organism, one can use just this surface polysaccharides per se. So they act as the immunogens, they activate immune cells, and you have an immune response, which is what is required by a vaccine. Now, say for example, the vaccine that is available for Streptococcus pneumoniae is made up of 23 such different antigenic polysaccharides. So this is actually mimicking a whole organism. So if on the surface of the organism you have 23 antigenic peptides, instead of having the organism with the 23 antigenic peptides, if you only have the 23 antigenic peptides, you are still mimicking the immunogenicity of the whole organism per se. So as you can see over here, this is specifically just polysaccharides, macromolecules that is forming a vaccine. Now, there are a few things that have been observed with polysaccharides as vaccines. Number one, what has been observed is that a polysaccharide can definitely be recognized by a B cell receptor, but what has been observed is that these B cell receptors, although they can differentiate to form a plasma cell and produce antibodies, and the antibodies are generally Ig2 and IgM type, that is because it is not activating a T helper cell. If it activates a Th2 cell type, then you would also have memory B cells being formed. Now, if you do not have memory B cells being formed, then you're not developing an immunogenic memory. And if you don't develop an immunologic memory, then you will not have lifelong protection. So therefore, that is a limitation that the polysaccharides has, that is they are not able to activate T helper cells, and because they are not able to activate T helper cells, you may have the plasma cells being formed, but the plasma cells do not undergo class switching. IgM is not converted to IgG1. IgG1 would be more specific for the particular antigen. So this is an observation, and therefore, although the immunogenicity is there, it does not have the power to develop immunologic memory, and that is something that is not good if you consider a vaccine per se. You need a vaccine which is going to create an immunologic memory. So therefore, there is a solution to that has been found. The conjugate vaccines can be more effective. That means that if you have the polysaccharides conjugated to an adjuvant, then the vaccine will be more efficient or more effective. So here, say for example, you're having the polysaccharide conjugated to a carrier protein. Now when you have the B cell taking up the carrier protein along with the polysaccharide, the carrier protein is going to get broken down into smaller pieces and can be exposed on the surface through MHC class 1. So as it does for the carrier protein, the polysaccharide also will get a kind of exposed, and therefore you would have a T cell getting activated. Now this is something that we want. We want that the T cells get activated. So now what you would definitely have is that a polysaccharide specific plasma cell producing the IgG1 and Ig3. So you can see that instead of IgM, you now have IgG. So there is a class switching, plus memory is also being formed. So example of this is Haemophilus influenza B vaccine, which is a conjugate of capsular polysaccharide of the HIB and the tetanus toxoid. So they have used the tetanus toxoid, which is a protein. So together, you will in fact get immunization for both Haemophilus influenza as well as tetanus toxoid. So this becomes more immunogenic because the carrier is now also having a protein and it can elicit the activation of a T cell. So it enables class switching from IgM to IgG, and that is why it has a better efficacy. Then one uses also toxoids as vaccines. So what are toxoids? It's basically that there are lots of bacteria that are able to release what is called as exotoxins. And these exotoxins are something that are able to give the diseases for the symptom. So if these exotoxins are taken inactivated, then you have the formation of toxoids, and when these are injected into the person, then the person is going to elicit an immune response. So generally what is done is that the seed stock is taken, you have the culture formed, it is scaled up, so you would have a large amount of cells producing the exotoxins, and those exotoxins will then be purified and converted to toxoids. So these toxoids can be used as vaccines. So these toxins definitely need to be inactivated, or else they are going to be harmful, pathogenic. So, but it has to be inactivated in such a way that the structure is maintained. That means its virulence or its pathogenicity is decreased, but its structure is maintained. To get large amounts of the toxins, the exotoxin genes are now cloned into bacteria that multiply fast. So if large amounts of toxins have to be obtained, then what is done is that the toxin gene is put into a bacteria which grows very fast. So with every division of the bacteria, you will get that many amount of toxins being produced. So the amount of toxins produced would be high, and then those toxins can be purified and inactivated to get toxoids. So the toxoids will induce anti-toxoid antibody formation. For example, diphtheria toxin, tetanus toxin. So in fact, one of the vaccines that is given to children is what is the DPT. So DPT stands for diphtheria pertussis toxin, pertussis tetanus. So all the three are actually toxoids. And so when a vaccine with all the three toxoids are used, then the child is getting immunized against diphtheria, it is getting immunized against tetanus as well as pertussis. Now, recombinant subunit vaccines is another way by which one can get purified macromolecules as vaccines. So a particular surface molecule that is present on a bacteria or a virus or a protozoa can be used as a vaccine because they are antigenic determinants. They are the epitopes. So say for example, one uses hepatitis B surface antigen, and this using EcoR1 is introduced in a plasmid. The plasmid is basically a yeast vector. So therefore, once it is cloned into a yeast vector and it is transformed into the yeast cells are cultured and one gets a high amount of the HBS antigen. So that can be purified from the cell culture and used as the vaccine. So it is less immunogenic because you're considering only one surface molecule, but when you use it with an adjuvant, the efficacy increases. So booster doses may be needed for long-term protection because its immunogenicity is less. But then instead of using the hepatitis B virus itself, it is safer to use hepatitis B virus antigen only that one, the surface antigen only. So this has proved to be much safer than using the hepatitis B virus itself. Now, one also uses synthetic peptides. So this was again a concept that was built up, suggesting that synthetic peptides are equivalent to those peptides that are being exposed on the surface through MHC class 2. So we all know that you have an antigenic peptide on the MHC class 2, the T cell receptor is going to recognize it, and then the T cell gets activated, proliferated, and it gets differentiated and gives rise to either cell mediated immunity or humoral mediated immunity. So instead of using again huge macromolecules, one can use just small antigenic peptides that have been understood to be immunogenic per se. So for example, you have a vaccine given. These are soluble antigens, and these are recognized by the PRRs present on the macrophages or dendritic cells. These antigens are then broken down into smaller fragments, and these antigenic peptides are now presented on the surface through MHC2, and that is recognized by the naive CD4 plus T cells, which then gets activated to either activate B cells or to activate T cells to form CTLs. So as mentioned, either the humoral immunity is going to be enhanced or the cell mediated immunity is going to be enhanced. But the point that we are trying to make is that it is this peptide that is activating the naive CD4 plus cells, and so why not just use that? So these presented peptides are either B cell epitopes or they are T cell epitopes and are able to elicit an immune response. Now, there are a lot of epitope designs that have been made to ensure that they are stable when they are injected, and these epitopes are able to elicit an immune response. So again, immunogenicity is there, but it is not that great because it's just a molecule, and so one needs adjuvants to elicit a higher response. So this is something that has been observed with macromolecules as vaccines. You would either need booster doses or you would need to use it as with an adjuvant. Then you also have multivalent subunit vaccines. So for example, we said that you can use a T cell epitope. If you use a T cell epitope, it is going to elicit the cell mediated immunity. If you use B cell epitope, you can elicit the humoral immunity. But what if you use both? Okay, so say you have a solid matrix on which is present monoclonal antibodies, and these monoclonal antibodies are specific for the T cell epitope and the B cell epitope. So you will have, as you can see, the B cell epitopes and the T cell epitopes bound to on the surface. Now, this is mimicking an organism, okay, but it is not a live organism, and so this can elicit now an immune response. So these are what is called as multivalent subunit vaccines, where you have more than one epitopes present on the surface, and those can be recognized, and the immune cells can get activated and produce an immune response. Now, one can also use multivalent subunits in the form of micelles or in the form of liposomes. So these can be taken up by the cell. These can be, degraded into smaller peptides and then expressed on the surface through MHC. So Again, these are therefore multivalent subunit vaccines that can be used. So in the liposome, for example, you have several different types of antigens being present. So again, this is kind of mimicking a whole organism, but it is not a whole organism. It is just mimicking a whole organism. So let us make the conclusions. As use of whole organism comes with risks of infection, macromolecules of the organisms can be used as safe vaccines. Bacterial surface polysaccharides bound to carrier proteins have been found to be immunogenic and also develop memory. Exotoxins produced by bacteria can be inactivated to form toxoids. Genes of surface molecules can be cloned and produced in vitro, and those products can be used as vaccines. Small synthetic peptides, which would be B cell or T cell epitopes, can be used with an adjuvant as a highly specific vaccine. So many vaccines have been designed considering only a very specific macromolecule to elicit an immune response and memory. This makes it more safe than live attenuated vaccines, and the production can be increased by cloning. Thank you.